Okay, so welcome back. We're in Bio 212, and what we're looking at, of course, is Chapter 22. And a moment ago, I left us off at the structure of the lymph node. And what I'd like to do is move us now, just to review quickly. Here's a capsule, and you have a subcapsular space, and you have these trabecular structures that are traveling through. And you have a cortex that has different populations of cells in it, depending on how deep you are inside that cortex. And then inside the medulla, you have a different population of cells here. And more importantly, not that you don't need to know what these cells are, you definitely do. You need to understand that fluid, that interstitial fluid, that is now lymphatic fluid, is now bathing over all of these cells as it exits towards that lymphatic trunk, towards the cisterna chyli. Okay, well, what does this actually look like? Here is a dissected lymph node. You can see there are many afferent vessels traveling in and a few efferent vessels traveling out. This is what it looks like inside your body, and these are the things that swell when you get a cold. And it's important for lab to remember you have submandibular, cervical, axillary, inguinal lymph nodes, and each of these will swell, and that's because that's where peripheral infections will localize inside this space. And you can see there's that subcapsular sinus, there's a trabecular sinus and structure here, and as you move down to the medulla, that fluid comes in and bathes over all of these cells. Now, it's slightly different than the spleen, and what I want you to see here inside the spleen is here's an artist's rendering of it, and you have the splenic artery traveling in, and you can see where the blood flow passes in, and you have that red pulp and the white pulp. Remember, the white pulp inside lab looked more red, and the red pulp looked more white. There's something significant in that when you actually get into types of cells, but beyond that, B cells travel here. In fact, that's probably just what I would write there, okay, primarily. If we were to think about the distinction here, B cells are traveling there, not necessarily mature, but they've left the bone marrow. Mature, now they're looking for a docking place till they diffuse through the body. And that's what you're thinking about when you're looking at this. Now, lymphatic nodules, similar to lymph nodes, are scattered throughout the body. They do not have a capsule, and they're traveling inside the lamina propria. Remember where the lamina propria is. It's the basement membrane for all mucosal membranes, right? So let's put a bold on that to make sure you get that. Okay. In the gastrointestinal tract, urinary tract, reproductive tract, and respiratory pathways. So everywhere your body needs to protect itself from materials that could be ingested or placed inside of it, right? Inhaled or ejaculated or and you've ingested. Okay. All of these are potential pathogens that the body needs to be able to detect. And you put a lymph nodule there, and that lymph nodule does not have a capsule. Inside your gastrointestinal tract, you can also have these things that are called mucosal associated lymphatic tissues, or MALT tissues. The further you go along in these studies, you're going to see more and more of those. What we want to do is we want to switch now to, if that was the basic structure of your lymphatic system, what types of immunity do we actually have? And I'm not talking, what is that stupid television program where you go to Immunity Circle or whatever it is, you get the necklace. That's not what I'm talking about. I'm talking about something that's called either innate immunity or adaptive immunity. And we'll start with innate immunity, where the body refers to, I should say, or responds to invasion of pathogens and their toxins that are non-specific, I think would be the easiest way to think about this, right? Innate immunity. I can actually get this to type anything into it. 2 lines of defense that are non-specific. Right? That's the important part here. And if you think about that, that makes sense. Your skin is non-specific to the type or number of bacteria that are on it. Inside your body, you're going to have innate immunity to bacteria that are found inside the fluids of your body. You'll see how this actually happens as you go along. But what's important, of course, is how does it do it? You actually have mechanical defenses. Mechanical, let's underline that to make sure you get it. Whoops, not that. Let's go Control Z. Let's go Control Z again. Mechanical defenses. Let's do that. Let's do that and see what happens. There we go. Fantastic. Okay. Skin, mucous membranes, tears, saliva, the cilia, the epiglottis, flow of urine out of your body, defecating and vomiting. These all get rid of materials inside of your body that could otherwise harm you. Chemical defenses, the different types of sweat you make, the sebum, what's inside of them, the enzyme, the lysozyme, okay? and or gastric juices. They will all destroy materials by changing their three-dimensional structure. Remember, gastric juices, hydrogen proton rich. Enzymes break down specific amino acids. And inside of this, you actually have antibacterial materials that are part of the sebum itself. But this is going to be the most obvious. Inside your body, you have cells, this phagocytic cell right here, right? This looks like, it's actually killing that microbe. And it's kind of different than what I draw in class, right? Where in class I draw something that looks like this, rah, 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 right? You got the rah, rah, rah mouth there, and you got the big eye, and you got the hairy, furry fur brow, and here's the poor bacteria that's saying, help me, right? But take a look what's happening here. There's the microbe, there's the phagocytic cell, it sends out the pseudopods, it ingests it, and it's killed it. Phagocytosis will remove these. You have white blood cells, remember from chapter 19, these are natural killer cells, these are lymphocytes, okay? So those lymphocytes, L-Y-M-P-H, lymphocytes are running around the body. Their job, of course, is to kill. And what they do is they kill proteins that are improperly made inside the cell, expressed at the cell's surface, and destroy the cell that's making the wrong protein. That's really the way it works. I'll explain it more inside class, but generally speaking, that's how it works. Inflammation, of course, is to increase the temperature inside an area by perfusing the tissue with more warmth, changing the three-dimensional structure. And of course, the nth extreme of that is a fever. Okay, and fevers increase the temperature inside your body, so you kill primarily bacteria that can't live in that range, but also think about what a virus is. It's sort of very unstable structure that if you warm to a specific temperature, you can get rid of it. How does this work? Okay, well, you can actually see here, get out of that, you can actually see cells move by chemotaxis to where the microbe is. They adhere to it. They extend those pseudopods I was just telling you about. They ingest it. They put it inside a vesicle that's called the phagosome. The phagosome fuses with the lysosome, and now you actually destroy it. Okay? And that the destruction, you can actually see here in that cartoon what's going to happen to it. That's its fate. It's going to end up, the residual bodies are going to be exocytosed out of the cell. Okay? That's what's going to happen to it. As you have large numbers of these cells moving to an area, guess what's going to happen? You're going to actually have inflammation inside that site. That's why every time you've ever gotten a cut, it's gotten red and what we call angry, right? You can actually see using emigration, which of course is just slipping between the endothelial cells here because they've been vasodilated. You actually have different types of white blood cells moving into this space to now actually move towards the microbe and destroy the microbe. Notice these are agranular white blood cells and these are granular white blood cells. Okay. Inside this presentation, I have table 22.1 as a summary of the innate defenses, and then the next table, of course, is going to be the summary of their functions, the second line of internal defenses. I'm only bringing this up because I'm going to develop it more inside class, but interferons actually protect your cells themselves from viral infection. And notice what they're actually paying attention to. They're paying attention. Whoa, what did I just do there? Sorry about that. Okay? They're trying to pay attention to uninfected host cells. Okay? Likewise, natural killer cells release granules that contain specific enzymes that then are going to be killed because of what they're doing, meaning they're improperly making materials inside the cell. Okay? As we segue now into adaptive immunity, what you need to understand is this is very specific. And the concept is actually really hard to teach. The way Mother Nature has designed your immune system is she rearranges the genome of each of these individual cells to produce proteins. And remember, all proteins go, let's think about this for a second here, right? Let's see if I can get this to go. Remember, the rules for how cells function. It's DNA goes to RNA, goes to protein, right? P will stand for protein here because we're going to run out of space. Protein, right? And that means the receptor to identify the specific invading agent has to actually have its own genetic information to make that specific receptor. And the way to think about this is you actually have T cells down here, T cells that have unique receptors to any possible pathogen. And pathogen is identified because it is specifically an antigen to the immune system. Whereas antibody-mediated are B cells that actually have receptors to pathogens. And the important difference here, of course, is that response to that antigen is going to cause the secretion of an antibody. And you'll see how and why in a few minutes. Okay? So what is an antigen? Okay? Antigens and antigen receptors define what we call immunogenicity, and they are reactive to either it's actually the easiest way to put this, of course, is the parts of the microbes that actually act as an antigen, and they are called epitopes. That's what I want you to pay attention to here. Individual epitopes are going to cause the activation of the receptors so that something interesting is going to happen here. So how does it all happen? I'm going to give you this sort of like screenshot. And it's kind of funny that the sort of green area here inside the image looks like a old school television, right? What's actually going on here? Well, this green bordered area is supposed to suggest to you that things happen inside the lymphatic system as a result of mature B cells moving into it and mature T cells moving into it. Notice those mature T cells left the thymus, mature B cells left the bone marrow, and they're now going to occupy different parts in it. And one way to think about this is a lymph node, but it doesn't have to be a lymph node. It could be a lymphatic nodule, so on and so forth. I'm going to go over this figure in a lot more detail inside class, but here's what I want you to see. Not only are they lymphocytes inside the body, you have specific populations of them. Cytotoxic T cells, helper T cells, and B cells. Remember I told you a moment ago where they come from. They each have different types of antigen receptors on their cell surface, and they're unique for each one of these. Notice how the receptors just look a little bit different from one another. We're going to go into how those are all activated, but the idea, of course, is you have the central regulator cell population. These are called helper T cells, and they activate cell-specific responses or cells that release antibody into the plasma of your blood to identify antigens or epitopes of antigens. Okay? That's generally speaking how it works. There's a lot more to it than this, but at the same time, I don't want to overwhelm you with all the details. Take a look at those receptors, and now take a look at this figure here. This is an antigen, and an antigen can be broken down in a million ways to Sunday, but whatever's left behind is going to create small, unique pieces that are going to identify that antigen as unique. These are the pieces that have to occupy or activate those unique receptors. And how it happens is primarily, the basic mechanism is named affinity. Okay, that's actually how you do this. Put in a text box here. So let's go like this. Epitopes. Epitopes are discovered by affinity. Affinity to their unique unique T cell and B cell. Okay? No lie there. Okay, that means if you start wrapping your brain around this idea, there are unique T cells and B cells to essentially any antigen that you may encounter inside your body. Obviously, as you age, that may change, but generally speaking, it's the truth. How do we actually process them? For us to actually identify them, B cells and T cells must be able to recognize that, and it's going to look at that fluid that's passing over its cell surface. That's really all it is. The fluid bathes over the cells. The receptor goes, oh, look what I found. And the antigen's like, oh, sugar pops. Okay? But once it's done that, then you actually activate the pathways for antigen processing. What's going to happen is, in class, I'm going to draw out for you what these MHC molecules are, but essentially, antigen, major histocompatibility complexes, are going to present to T cells and B cells. And I'll show you how in a moment when you're looking at this figure. I just want to go through two pathways and then we're going to move on to the next set of slides, right? So if a material is brought into a cell, it's digested, MHC2 molecules bind to that unique antigen and then express it on the surface of the cell. You're presenting it then. It's not the only way we can do this. Imagine, if you will, a cell's been virally infected. You now have an endogenous protein. It's now broken down through internal cellular mechanisms. It's now presented at the cell surface, here using MHC1, but this is still an antigen-presenting cell. So what's going to happen, of course, is T cells, B cells will see these antigens and they will then become activated, which is a good place for us to stop, because in the background of all of this isn't just what's going on inside these cells. We have to think about what the hormones are that are actually helping to regulate this entire process. Okay, see you in a minute.